Every fruit contains antioxidants. That is not a marketing claim, but a nutritional fact rooted in the basic biology of plants, which produce antioxidant compounds as part of their own defense systems against environmental stress. When we eat those plants, we absorb those compounds and benefit from the same protective chemistry. But within the fruit category, some options are substantially more concentrated sources of antioxidants than others, and understanding which fruits stand out and why helps translate a broad health concept into specific, actionable food choices. What antioxidants are actually doing? To understand why certain fruits are particularly valuable, it helps to understand what antioxidants do at a biological level. The body continuously produces free radicals as byproducts of normal metabolism, and exposure to environmental factors like ultraviolet radiation, air pollution, and cigarette smoke adds to this burden. Free radicals are unstable molecules that cause oxidative damage by stealing electrons from nearby cells, proteins, and DNA. Antioxidants neutralize free radicals by donating electrons without becoming destabilized themselves, interrupting the chain reaction of cellular damage before it accumulates into the chronic inflammation that underlies heart disease, cancer, neurodegeneration, and accelerated aging. Different classes of antioxidants operate through different mechanisms and protect different types of tissue, which is one of the reasons that dietary variety matters. A single antioxidant compound, even a highly potent one, cannot replicate the protective breadth of consuming a range of fruits and vegetables with diverse phytochemical profiles. Blueberries. Blueberries consistently rank among the highest antioxidant foods measured by ORAC scoring, a standardized assessment of antioxidant capacity, and their primary active compounds are anthocyanins, the pigments responsible for their deep blue-purple color. Anthocyanins are a class of flavonoids with potent anti-inflammatory properties, and research has linked regular blueberry consumption to measurable improvements in cardiovascular health markers, including blood pressure, arterial flexibility, and LDL cholesterol oxidation. There is also a growing body of research associating anthocyanin intake with cognitive protection, with studies showing associations between regular berry consumption and slower rates of cognitive decline in older adults. Blueberries are also a meaningful source of vitamin C, a water-soluble antioxidant that works through different pathways than anthocyanins, supporting immune function, collagen synthesis, and direct neutralization of free radicals in aqueous environments throughout the body. The combination of multiple antioxidant classes in a single food generally produces synergistic effects greater than any single isolated compound would deliver. Oranges. Oranges are best known as a vitamin C source, and their vitamin C content is genuinely significant, with a single medium orange providing the full daily recommended intake for most adults. But oranges also contain flavonoids, most notably hesperidin, and carotenoids, both of which provide antioxidant activity through distinct mechanisms. Hesperidin has been studied for its effects on blood vessel function and blood pressure, with research suggesting it supports arterial dilation and reduces markers of endothelial inflammation. Carotenoids, which are responsible for the orange pigmentation, contribute to eye health and immune function in addition to their antioxidant properties. A critical point about oranges is that whole fruit provides substantially more benefit than juice. The fiber in whole oranges slows sugar absorption, supports digestive health, and interacts with the flavonoid compounds during digestion in ways that juice cannot replicate. Research has also found that the antioxidant compounds in whole citrus act synergistically, meaning the combination of compounds in the complete fruit is more protective than isolated vitamin C alone. Blackberries. Blackberries share the anthocyanin richness of blueberries, but add a particularly notable concentration of ellagic acid and ellagitannins, a class of polyphenols with well-researched anti-inflammatory and potentially anti-carcinogenic properties. Ellagic acid has been studied for its role in supporting detoxification pathways and inhibiting the proliferation of certain cancer cell lines in laboratory research, though translating these findings to specific human health claims requires considerably more clinical evidence. Blackberries also contain resveratrol and quercetin, two additional flavonoids associated with cardiovascular protection and anti-inflammatory activity. At roughly 8 grams of fiber per cup, blackberries are one of the higher fiber fruits available. And this fiber plays an important role in gut health beyond digestion. Gut bacteria ferment the polyphenols in blackberries into metabolically active compounds that have systemic effects throughout the body, meaning the actual antioxidant benefit of blackberries depends in part on the composition of your gut microbiome. A diverse fiber-fed microbiome converts these polyphenols more efficiently into beneficial metabolites. Red grapes. Red grapes are among the most concentrated food sources of resveratrol, a stillbean polyphenol that has attracted substantial scientific attention for its cardiovascular and longevity-related properties. Resveratrol activates cellular defense pathways, reduces LDL oxidation, inhibits platelet aggregation that contributes to blood clot formation, and has demonstrated blood pressure-lowering effects in clinical research. The compound is particularly concentrated in grape skins and seeds rather than the flesh, which means whole grapes provide more resveratrol than grape juice, and seeded varieties provide more than seedless ones. Beyond resveratrol, red grapes contain proanthocyanidins in their seeds, ellagic acid, quercetin, and camphorol, making them one of the more phytochemically diverse fruits available. The concentrated antioxidant profile of red wine relative to white wine is directly attributable to the extended skin contact during red wine fermentation, which extracts these compounds into the final product. Whole red grapes, however, provide these benefits alongside fiber and without the accompanying alcohol. Pomegranate. Pomegranates contain a unique set of antioxidant compounds called punicalagins and puniculins, which are ellagitannins not found in meaningful quantities in other commonly consumed fruits. These compounds are converted by gut bacteria into urolithin A, a metabolite that has been shown in research to support mitophagy, the cellular process of clearing out damaged mitochondria and replacing them with healthy ones. This makes pomegranate's antioxidant activity particularly relevant to cellular aging and metabolic health in ways that extend beyond simple free radical neutralization. Clinical research on pomegranate has documented reductions in blood pressure, decreases in LDL oxidation, and reductions in multiple inflammatory markers. The arils, the edible seeds surrounded by juice sacs, are preferable to pomegranate juice for the same reasons whole fruit generally outperforms juice. The fiber remains intact, the sugar is more slowly absorbed, and the full phytochemical profile is preserved. The principle behind the list. What these 5 fruits share is high concentrations of specific antioxidant compounds with documented mechanisms of action and meaningful bodies of clinical research connecting their consumption to reduced chronic disease risk. What they also share is color. Anthocyanins produce deep blues, purples, and reds. Carotenoids produce oranges and yellows. Ellagitannins contribute to deep red and purple pigmentation. The connection between fruit color and antioxidant content is not coincidental, but reflects the same plant chemistry that produces both the pigment and the protective compounds. Including these fruits in a varied diet rich in vegetables, legumes, whole grains, and healthy fats provides the broadest possible antioxidant protection. No single fruit, regardless of its antioxidant density, substitutes for the collective benefit of genuine dietary diversity.